It is a great pleasure to be here. So after a long time. So I did my graduation and PG PhD from as an alumni from TNA only. So I left this college for 2008. And past 16 years I am working in Indian Institute of Rice Research as a senior scientist. So my area work already Madam mentioned. I am working exclusively for direct seeded rice. So that is my like abiotic. So now water is a major concern. So that is priority area. I am concentrating hybrids for abiotics. Today I am talking mostly overall what is the hybrid rice research going on in India, especially in our institute and some nodal institute like NRRI, 1 or 2 slides. So before going, so already you know some basic 3-4 slides I will go as a student. You have to refresh what is hybrid rice, why we are going for that. So little refreshment like that. Then I will go to the in-depth research what we are doing. So you know what is hybrid rice. So can you recall it? So how many are working in rice? So all are PhD, na? So I think MSc also include rice. I think most of them are familiar with the rice. So if you are making 2 crosses, if it is a diverse parents, so then we call the hybrids. But is it not possible for us to make every time emasculation and make crosses? That's why the term hybridized technology, if you make a 2 variety also, the output is called hybrid only. But when we come to a large scale production, we need a different method. There comes the CMS and all other concepts are there. I will be talking on that. So first generation offspring cross between 2 diverse parents we called as a hybrid. So why we need hybrid rice? Anybody? So we know that our population is now we are leading. We are the top one in the population. So to feed the nation, we need more to produce. So one option is hybrid rice. So what is the things? Because we need the productivity. So at least the hybrid rice, what we so far we are seen is 15 to 30% yield increase over the popular variety. So hybrids are popular because of the yield productivity and also the economic returns. That's why we are going for the hybrid rice. So how to measure heterosis? So you know the hybrid rice is working on the concept of heterosis. You know what is heterosis? Superiority of over the parents or the best variety. So how will you measure it? There are 3 types of heterosis are there now? Yes. So one is mid parent, better parent and best varietal. So normally we will use only the best varietal heterosis. For a promotion of the hybrid, we will see with the variety. It should be what our national system, I will come to that slide. National system we are doing is, it should be 10% better over the variety. And it should be 5% over the best hybrid, existing hybrid. So for example, if you are coming to Tamil Nadu already co-RH 4 and 5 is there. You are releasing a hybrid. It should suppress the already existing hybrid over 5%. For example, that if you are releasing a yearly hybrid, so it should suppress how much? That Co51, Co50, 10% over that popular variety. So then only it will be released. That 3-tier system they will be following. So in that this should follow that criteria and also quality. These all there only it will get released. So already we mentioned what is the need for hybrid rice. So you know that these are all the recurrent problem which is we are encountered every year. One is the population. There is no chance there will be a decrease in population. So land area also it is going on decreasing. So next one is pest and disease, water shortage. So these are all the problems are there. We have to produce more with less resource. Okay. So if you see, there are 2 chances are there. We cannot increase the land. So we have 44 million hectares of land is there. So what that is called horizontal expansion. We cannot go beyond that. So only what is a possible solution? So that is vertical expansion. Vertical expansion means increase in yield. So only chance is through varietal improvement for with more yield. Another one is possible is hybrid rice. This is a feasible technology. We can go ahead with more yield. So how to exploit heterosis? Any idea? So this you might have heard na, in your MSc or PhD coursework. So there are, if you make cross with this, you know that the subspecies of rice, indica, so Javanica, Japonica and indica. So if you make with the intercross indica and Japonica, tropical Japonica especially, we will exploit more heterosis. This is what we have seen so far. And the only problem we encountered in if you are going for the Japonica is quality. So quality will be poor. So what we are doing is we are going more back crosses. So then we will improve the yield and simultaneously with the quality. How will you do? There are, you might have heard, there are two commercially available technology in hybrid. So what are all that? Commercial system. 2 line and 3 line. So which we are using more exploited? 3 line. Okay. So this picture shows you how you might have gone through this one coursework. So there are 3 systems are involved. CMS, okay, in CGMS system, A line, B line and R line. So if you go to the 2-line breeding, you no need separately A line and B line. So because of temperature control, it will act as a line and B line. Okay. So this, you might have seen which cytoplasm we are using. Wild abortive. So there are so many cytoplasms are there identified that are theoretical and some are not in practical use. So wild abortive is a widely used cytoplasm which is identified from the Oresa rufipocan as a natural mutant. So like another one, Honglian, BTCMS, these are all we don't have good restorers. So that is the main problem. We cannot exploit hybrids from that kind of CMS lines. So 2-line, as I already told, it is based on TGMS. What I have mentioned here is based on temperature control. So if you increase in temperature above 24 degrees C, so it will act as a sterile. So it will be act as a CMS line. So male sterile line. If there is a decrease in temperature, so below 24, it will act as a maintainer line. So here in Tamil Nadu, which place we are doing this 2-line hybrid? Gudalur. So this is a mechanism where the temperature control, it depends. But why it is not much successful? At any reason? Because our environment is not consistent. Suddenly there is a fluctuation in temperature during, especially during the flowering. So then it will revert back to the maintainer. So it will affect the seed production. That's why what is we are doing is we want to stabilize that line even 2-3 degrees centigrade. If it is a less or more, then and it should be stable. Otherwise, this technology it will not be visible. But in case of China, they are exploiting. This is a major technology. They do 50% of their area is in 2-line hybrid. So this is a just so far I have what I told you, just a glimpse how you want to refresh it. What about a hybrid rice? So now what the network we are doing, how the technology we are taking forward. We Indian Institute of Rice Research, Hyderabad, we are the nodal organization. We are looking for the research. AICRP, you have heard, no? All India Coordinator Rice Improvement Program. There around 45 funded center and more than 100 voluntary centers are involved for a varietal program. So in that, these 10 center, 11 center public centers are involved exclusively for hybrids. Coimbatore is one of the center, like Mandya and Karnataka. So NRRI, that Cuttack center is there. Then Maruteri from Andhra Pradesh. So Karjat in for Western country, Western India. So Karjat is there in Maharashtra, North in New Delhi. So like that, every zone we have one center for hybrid rice research. So that are looking overall research and the seed production in India. So other than that, 45 funded and more than 100 voluntary centers are involved in testing. Yeah, this slide is see in whole country or in a whole world how we are and China. If you see the first two, in India we see the area is 43 points or 44 million hectares. If you see China, it is 30. But if you see the rice production and the hybrid rice adaptation, it is comparatively higher. It is 50% area is in that hybrid rice only. Especially two-line hybrid is more in China. So, but in case of India, we have around 6% only of the area. So our still we have way to go for adaptation. So if you see Vietnam, Bangladesh, Philippines, so these are all merged area. They have doing at least 4 to 5%. So major contributor is from India and China. So far in India, 152 rice hybrids are released. So if you see varieties, at least 1,300 varieties are there. So in that 150 only from hybrids. So if you see in that public sector how much and the central state release, if you see central release means it is more than one state it is released. We call it a central release. If it is released for Tamil Nadu and Andhra, then we will call Central Varietal Release Committee that will identify. It will be tested national level, then it will get released. Otherwise, state varietal release means it is tested within Tamil Nadu only, then it will be released. So if we see central release 105, rest are from state release. So already I told the area is 3.5 million hectares, so it is 6%. So if we go on to the research area, what we are doing for the improvement of hybrid rice. So these are all some of the areas one by one we can see. So one is diversification. So as I told, there are 5, 6 CMS are available, or the cytoplasm we are using, but which is commercially is AA only. But if you have any chance of outbreak of disease or any problem is there, then this will hinder the whole production system. So we are doing some of the diversification. One, we are seeing the outcrossing. Because if we make A into R or A into B, we will get 30% seed set only, or 40% or 50%. So to increase more seed set in the A line, so what we have to do? The outcrossing should be very good. What is outcrossing? Any idea? Seed set. So A line, when the pollen from the B or R line, if it falls, there should be more of seed production. Seed set should be more. For example, their sigma exertion should be good. Lemma palea should open for more time. So these traits are not there in case of CMS, what we call a line, then it will not be a good CMS line. So we are improving that period of lemma palea opening or sigma exertion, double or single sided sigma exertion, then this will facilitate more seed production. So this is one area we want to diversify. So we are working on that. So another one, next one is panicle exertion. If you see, if you want to differentiate A line and B line, so A line, B line are isogenic line. So only difference is sterility. So remaining all it looks like similar, alike. So however, there will be still some problem is panicle exertion. So in case of CMS line, we will not get complete exertion. It will not be 100%. So that also morphological marker to identification, differentiate AL and B line, there is a contamination, but it will reduce the seed set. So what we are doing is we are putting some genes, EUI genes, that will elongate the internodes, so that will facilitate more better panicle exertion. That is the right. We will get at least 75 to 80% only as of now. So we are expecting more, 90, 95. So these are all some I have mentioned, that is Gambiaca, Desi base, the RTN is Ratnagiri, which is in Maharashtra. They are doing some of the work on that. But unfortunately, whatever CMS with the new cytoplasm, unable to get the restorers. That is a major hindrance in diversification. So what I told the stigma exertion, what we are doing is Longistaminata, which is a wild rice. So we are going for a AB QTL, that is like advanced backcross QTL analysis they did. So here that stigma, which are all the lines, which are all the spikelets having exerted stigma, we are trying to capture that and we are advancing those generations. So this will help in outcrossing. This is the one work. Next one is what we are doing is the CMS, whatever already we have. So we want to improve it. Still, whatever thing is there, we have to improve for the. Now we are improved for BLB and for the POP1. So POP1 is for the phosphorus usage. If you see India, more than 70% we are importing from the other countries, phosphatic fertilizers. So this popon gene, which will help in proliferous root system, so that will help to get the maximum nutrients from the soil. So this gene we are introgressing into the variety also simultaneously, like in B line or restorer line. So we have advanced population. So with this we have did the experiments that it will reduce the use of phosphatic fertilizer. This is one thing. So if we come to the WA CMS, so what is the gene involved? What is the mechanism? So this we have to see. The WM is wild abortive. So this trait is in cytoplasm or in, so mitochondria in the cytoplasm, this controls the sterility. So there are some candidate genes we have identified like WA352. So this this is initially identified in the rofipogon. Okay. So we did the marker analysis and we found that this is the thing so which can give 100% sterility. Okay. So what we did is, normally if you go grow test, what is grow test? So you want to see the purity of the material. So you will raise panicle to row. Okay. Then one set we will do, another set back after 20 days or 30 days, you will grow it, rest of the material of that panicle. So once it is in flowering, you will see how much mixture is there. Okay. So with the help of our markers, we have developed for this WACMS, if there is a mixture within the CMS line. Okay. So each CMS has base pair variation is there in the WA. So what we do, if you grow test, it will be time consuming. It will take more time. So in seedling stage only, we will do the marker analysis. And with the base pair difference, for example, number 16. So what we'll do, that seed lot we will discard it. Okay. So this method, we can see how much impurity is there in the CMS line. So then we can go good. So this is a method what we can go for the purity identification. You see, this is this slide will give you a better example. So we have series of sort of CMS line we have. So if you want to identify which has the impure, which are differ based on the difference in the base pairs difference, then we can differentiate which is the seed lot as the impurity. Okay, within the A line. Then in 3-line system, we have seen so far the CMS. So how the sterility will be restored? We need the fertility line. That is what we call the R line. So R line, there are two genes are involved. One is RF3, another one is RF4. So RF4 is a major gene, so which controls the fertility of the by suppressing the PPR protein, that is pentatrichopeptide, that is which is involved in suppressing ability of the CMS line. Okay, so that will nullify the effect of the CMS. Then seed will be produced in the F1, what we call F1s. So if you make cross CMS into a R line, so these two genes are involved. If you see RF4, which is a major gene, at least more than 80 to 90% seed setting is because of RF4. If two combination there, we will get the 100%. So in this, what we did is we identified within the RF4 there are putative genes. So that is what we call pentatocopeptide PPR we called. So those proteins will help in restoring ability of the CMS line. So in that we found that PPR9, which is a major thing, it is fine map. We have now gene-based markers, so that will help in identification of the series of restorer line in a population. Then we will use in the our hybrid. So this also can be utilized in case of identification of in a hybrid we are giving. There is a, for example, if there is impurity is there also. First one is CMS line, base pair difference will be there. Next one is restorer line. Okay, these are all restorer lines. Okay. If you see this one, these are all restorers. The down one, if you see CMS line, these are all restored. There is a base pair difference. So based on the base pair difference, these are all the genotype. It is a restorer or non-restorer we will identify. Okay. So it is a base pair of 54 base pair difference, 47 base pair difference. This marker difference is there. Based on that, we will identify these are all restored. We no need to make test crosses. We no need to time consuming. So with the based on markers, we utilize for the crossing. So it is heterotic or not that we have to do later. First, it is a restore or not, we have to identify with the markers. That is a company or somebody is giving a hybrid. So once you raise what we call grow test, these are all time consuming. Like CMS, what we did here also, we can with these markers, we can do it in with the help of marker. If you see the first one, there are some impurities. So in hybrid, you will get heterozygous band. We will get both A line and R line bands. It is like a, what is that? Is it dominant or codominant? So wherever we have difference, those all we will, from the lot, we will discard it. Okay. So we will take random lots. So when somebody is applying the seed, so we will do this analysis, then we will get the results. So if we go grow test, you need at least 2 seasons. One season if you raise, then result based on the result, next season only you can give the seed. But with the help of marker, you no need to wait for that. So you can with the seed or the seedling also, you can extract the DNA and we can do the in case of RF3. This is another gene which is not a major gene, it is a minor, but it combination of these two genes are essentially for the restoring the fertility. If you see here, so we have now narrowed down RF3, that RMS SF25 on -5. This marker can distinguish the restorer and non-restorers. So we narrowed out, these are also gene-specific markers, which can exactly we can identify the restorer with the possessing RF3. So this is one example of what we did. So here you see, these are all the restorer populations. So wherever A line also there, B line, A line, B line, and R line, non-restorers also we can identify. These are all non-restorers. These are all restorers. So based on the base pair difference, so we can identify the genotypes. So next one is what we need is S5. So when we are if it is in Indica, this problem is not there. When you are using a tropical Japonica line in your breeding program, this S5 plays a major role. This is a sterility factor. Okay. So when your CMS and on your R line you are making cross, if it is S5 neutral allele is there or S5 Japonica allele is there, S5 Indica. These three are there. If you have Japonica allele, then you will get more sterility. So you have to identify the genotype which is devoid of that. So this marker will help in exact identification of the S5N neutral allele. If you see the Indica, Japonica, there is base pair difference and wild compartment genotypes. So a genotype which is having S5 neutral allele always better to use in the breeding program when you are using a Japonica or some driver genotypes. Next one is you have read somewhere or the recurrent selections. You see what is the recurrent selections? You also read about reciprocal recurrent selection. Huh? So these are all what for that? Population improvement. So you have to improve the diversity. So you increase the diversity of the genotypes. So population improvement plays a major role. So in case of hybrids, what we are doing is we have a population for restorer and maintainer, like a CMS line. So we will make open pollination. If you see genetic males, IR36 population, we have a restorer population. So once we make cross with that, with the diverse donors, we will get 1:1. What is 1:1? If you have two plants, one plant will be sterile, another one will be fertile. So we'll use the sterile line for the further crossing. Fertile one will select and grow separately. Okay. So similar way, every season we will do that. So we will cross with all the donor. F1s will be raised. F2. F2 all CMS line will be bulked. Okay. What the 1:1 ratio we are getting na? So whichever fertile will go separately panicle to row, that is a restorer. Whichever from CMS we are getting the seed, we will bulk it. So then once again next season also will rise. So next season also will cross with a different set of donors. So the genome will be mixed. So we will increase the diversity. So finally we will get the diverse material from that population. That is the purpose. That is the purpose of this one. So we will call each one as a one recurrent selection. So this process will go on repeating. This we are doing at least more than 25 years. So this is every overall coming, this they will be maintaining. So now the population from that is improving. From that, the best material we will take out and we will use in the hybrid program. So as we are talking about the two line, so these are all the new technology which can in future will improve the hybridized yield or sustainability, what we call. So one is two-line breeding. So far around 10 genes are identified for temperature male sterility. In the TMS5, which is a major one, this is a recessive gene, but we are using in majority of, if you see in China or India, this one is the major one. So what is the reason is, it will give the stable performance under the 24 degrees C. So now what we are doing is, you might have heard about the genome editing. Okay, so I think some lectures you might have gone through. So what we are doing, we are editing this gene, TMS5. So initially, some reports are there in China. So they got some new TGMS in their population. So same methodology we are using, we got a project on that. So if you see, China 71% having this TMS background. So now we are editing that TMS5. So whatever lines we will be getting now, genome editor, like a mutants. So that will be evaluated in the like good or controlled temperature condition, how much sterility it will be we are getting. So when we will increase the temperature, decrease the temperature, how much stable it is there. So major purpose of this is, it should be stable. If there is a line which is sterile at 24, even there is a deviation in temperature for 1 hour, 2 hours, or if it comes to 21, 22 degrees C, it should not become fertile. That stability for 1 or 2 days should be there, because most of the lines what we produce is, even there is a weather, because suddenly there will be a rain, flowering time, there is a weather change for 4-5 hours, then it should not revert back to the fertility point. That is what. So most of the genes TMS 1 to 9, so 10, that are not stable. So for especially condition of India, the weather is not always stable, especially Rabi, Kharif season. Rabi we will have dry spell more, but in case of Kharif, you see today temperature is low. If there is a flowering, it should not come back. It will take both day and night temperature. It is average of that. 23, 24, we are not talking about the daylight temperature. It is taking about the both day and night, average of both. So it should be 24. So now we have M0 generation. So we are expecting some new TMS lines which is stable under this temperature. So next one opportunity what we are seeing is yield gene introgression. So we use the wild rice for a yield gene identification. So that we are putting into the hybridized background, like parental lines. So for GN1A, that is grain number we have identified. So that we are introgressing into the parental lines. Similarly, that wealthy farmers panicle we call, that is panicle branching. So it will increase the maximum panicle branches in the each panicle. So in hybrid, so that we can get more grain number and also strong culm. So which is more needed, especially when we are going for the direct seeded rice. So that we are seeing, and grain weight, that is GW5, GS2 grain size. So these are all genes. So how it will increase the heterosis? That is our study. Now we have final product, we are there. So we are nominating one by one. So there what we found is, So those will work because in hybrid it will be in heterozygous state, most of the things. You got now. So heterozygous state, that effect of the QTL or gene, it should be same, whatever it is in the homozygous state. So whatever our study we have done, there is no change. In heterozygous state also, it is giving the better yield. So as of now, we have products. So far, we're not at released, so that we have to done in the iCrypt system based on that. So next one is we know heterosis, because hybrid is heterosis phenomenon is a major thing. Why cannot we fix that heterosis? Why cannot identify the QTL which is responsible for the heterosis? So we taken KRS2, that is a popular hybrid. So based on the real population, you might have heard the recombinant inbred lines. So we identified some QTLs which gives better yield. So those lines we once again we crossed with the CMS lines. So we compared to the original KRS2, we found excellent yield increase. So those QTLs we are now in fine mapping stage. It will be utilized in the breeding program. So next one opportunity what we are seeing is heterotic gene pool. So what we call heterotic gene pool for maintainers and restorers. So what is the thing is, if you are taking, you have done the diversity studies. Diversity studies for purpose is to identify diverse parent and you have to make the crosses. So here are the same concept with the use of markers. Both morphological data we will see and the marker data. So based on the clusters we have identified, based on morphologically, these genotype fall into these clusters. And marker wise, these genotypes are in that. So we will make the crosses between the clusters, but not between the maintainer and restorer, within the pool. So crosses would not be different pool. Within the pool, we will make cross. We will see the how much heterosis is increasing, how much diversity we are bringing. So this is called what is the heterotic gene pool. So last season we got from all the nodal centers around 130 restorers we got. Even Coimbatore also we got. So we have preliminary studies. It is shows we have got 13 clusters. So both it is matching with the morphological data and marker data. So this is a preliminary study. We are going ahead. So these are all traits as of now we are improving. For biotic stress, if you see all the BLB genes, XA, all major genes we are as of now we are targeting XA 21, 33, 38. Likewise for BLAST, PI54 we are concentrating. BPH also we are concentrating 33, 31 and Galmage. So these are all the major biotic we are concentrating. Likewise abiotic, there are mostly are QTLs. Drought or there are not fine map. Saltal we are concentrating, PUP1. So these are all areas sub-1. We are some are in iCrypt trial. Already private companies are there little ahead of public. So already they have released. So Sabna, we have products on this. This is one success story I want to tell here. How in variety, you might have heard BPT 5204. That is a Samba Masuri. Like Ponni. So in Andhra we call Andhra Ponni we call that is BPT 520. We improved those all for these traits. So far we have released around dozens of varieties with these genes, BPT background. So for BLB also we have like improved Samba Masari we call, that is having BLB genes, 3 genes, and BPT with that DRR than 53 with 2 major genes and 3 minor genes. So likewise we have product, n number of products. Similarly, what we are doing is, though based on the success story, we are putting into the hybrids. As of now, we have improved for saltal, submergence, drought, and PUP1. So similarly for BLB also improved, BPH and other traits what I am mentioning here, like herbicide tolerance, anaerobic germination, heat. Heat also we have products. Low soil P also we have in finished products. So this will give a glimpse how all the traits we are putting in one background. Only thing is you need more population for evaluation. For one gene, how much population is there? Morning also there was a discussion. If you have less population, you will miss that gene. If you are going 5 genes, 6 genes, means the population size should be more than 1,000, 3,000, 4,000, then only you can able to identify. So all we are doing with the now strip markers, not SSR, Now we are outsourcing those material with the, like IRRI, they have developed a, markers for each trait. Around 130 genes they have now narrowed down. So we are giving our population for even for quality, all the biotic and abiotic tests they are done. So for around 400 samples, they are charging around 4 to 5 lakhs. So we are getting the product data within flowering stage. Based on that, we will identify the which genotype we have to forward it. So we will not wait up to next generation. So with this is the one success story from our institute, DRRH4. This is released especially for direct seeded condition. So this was released 2 years back and for the 4 states. And this is the first hybrid in the world for this situation. So there are so many works are going. So this is the first. This is when I joined that institute. We started this. It takes more than 10 to 15 years to bring a hybrid for this situation. The second hybrid from our institute, coastal salinated DRRH5. So this is a partnership program from our institute and IRRI. So this is also released for West Bengal, Goa, Andhra Pradesh. This is for coastal area. This is for the medium slender grain. So this is last year it was released. I was telling that for BLB. So this material, whatever for our institute we are doing, we will share that material. So whatever material we have finished, we will now only not use it. Whoever requests, we want the material with these genes. So one material which we shared, they made the hybrid. That is Navasari Institute, Gujarat. They released one hybrid with the BLB resistance. So if you see GRH2, this hybrid, they released with BLB resistance. So this is the one hybrid, two-line hybrid from IRI, PUSA GRH 56. It is from the two-line breeding system. So this also released last year only. Any idea from TNA any release two-line hybrid? This year, January, this was accepted. I was also one member in that SVRC committee. SAR was also there. So this was the first hybrid from this institute, TNAU. So released for two-line system. This is a major success. So this is the initial thing. So this is so many still has to come. So this is one nutshell how the seed produced, where it is grown. If you see the based on F1 hybrid produced, F1 seed, how much it is sold. Based on that only the area is calculated. Okay. If you see Telangana, they will call as a seed hub. Okay, especially Karimnagar and Mehboob Nagar, those area, they are only hybridized, they will grow. Only seed they will produce. A line, B line, A line, R line, that combination only will be there. They will produce the seed here, they will send to the Eastern India. So most of the hybrid grown is in Eastern India. If you see the picture, maybe next one also, 80% is in Eastern India. It includes Eastern UP, Bihar, Odisha, Jharkhand, Chhattisgarh. Though seed is produced here, southern, so grown is Eastern India. And the private contribution is more. So this hybridized program started in 1990s. Once you might have heard your long ping, when he identified a natural mutant from the Orisaru fever, that is CMS trait. He identified in 84s. Then that material came to us, Gensang 97. Then IRRI and other countries, they collaborated. They made the IR 5A025A. That is the first CMS entered into India. Based on that background only, so many hybrids, more than 50 to 60 hybrids are made. That CMS. So unfortunately, that CMS is developed in the background of basmati rice. So that's why we got the aroma in the hybrids. Initial days, hybrids were farmers not preferred in southern India because of that aroma. We don't prefer that aroma. So though it was in the background of the basmati, so that was not preferred. That first, the success was not taken into bigger effort. So now we don't have any aroma. So we totally eliminated that second generation. Now we have third generation hybrids. without aroma. So that 25A we are not using. So most of the company nobody is using. But first what the farmers or millers that the message conveyed it is wrongly went. So that's why many people are taking hybridized means it will be quality less. So that is not happening now, but still that sigma is there among the farmers or millers. The millers they want to take it in a lesser price. So this is hybrid means they are asking in a lesser price. That is why it is not going forward. Though nowadays hybrids are not like that. Still the stigma persists. That is a problem. See, mostly UP is a maximum thing in that eastern UP. So then if you see this side, Punjab, Maharashtra, Gujarat, those are all next level. They are cultivating some packets so that. But in case of southern India, AP, Telangana, Tamil Nadu, these all, the seed production is more, but the cultivation is less. What is the main reason is we are more preferring about the like pony, the fine grain varieties. Most of the hybrids are little bolder, early hybrids. Now we have a separate segment for medium slender. So like it should be like a BPT, it should be like pony. So we have separate trials. Whatever hybrids now around 10 to 12 hybrids are there with the medium slender grain. Not only the grain type, it should have a cooking quality. So most of the grain which is visible like a BPT, it will not have similar cooking quality. So that is a concern. So nowadays, with that generation hybrids, what we are producing is meeting the whatever the requirements of the farmers are the medium slender grain. Still, we have some constraints. So what are things? Marginal heterosis, 15 to 20% only we are exploited. Still, this is not sufficient. But in cases like a rice, self-pollinated crop, this is still a struggle. Or we have to go still better opportunity or better methodology to bring it the more heterosis into that. Next one is narrow genetic base, especially female. Female line means CMS line. Because if you can improve the restorer line very easily, but CMS line first you have to improve it, then you have to do the conversion program. So it is like a two-way work, and it takes more than 10 years. So there is no diverse CMS lines. Next one is consumer preference. So what I am telling now, quality. So India like country, so we need diverse preference of the eating quality is there. That's why the hybrids still not meeting the requirement of all the people. Still, we have to go a long way. Then, especially coastal areas, they need a long duration hybrid. Okay, like shallow lowland. So our hybrids mostly are early and medium segment. So long duration hybrids are still preferred. So yes, if you see the Swarna CR 1009, these are long duration varieties. So we don't have hybrid in that similar quality or similar durations. That is the one still we are. Then the last one is susceptible. That we have improved a lot. Still we have to go because one or two hybrids may not be sufficient. We need array of hybrids. Which one will click? So like varieties, we have 1,500 varieties released officially. But if you see the market or farmers, more than 40 to 50 only be there. So though released, but in case of hybrid, 150 only officially released. We have to release still more with the resistance and tolerance. Next one is higher seed cost. You know how much seed cost all are selling the F1 hybrid? What is the cost of the hybrid? It is more than 300 rupees per kg. So farmers, it is very difficult to take that one. Though we are recommending 6 to 15 kg only seed rate, still it is higher rate. So the returns they are getting, but still they are very not coming forward to take the hybrid seed. So next one is, as I told before, the millers. If it is hybrid, they will be automatically reducing the price. No, this is a hybrid, it may not be a good quality. So that notion they have, they are still telling hybrids are no. So that is another problem. So it has head race recovery is less. It will come, normally it should be above 55. If it is less than 55, it will not, they will not take. Even it is meeting because of that hybrid, the bird still some of the millers are not taking forward. This is initially we did with the help of IRRI, we got the CMS with the diverse cytoplasm. Unfortunately, we didn't got restorer for that. So for WA CMS, no, we used Minuta, used Nivara, Rufi, we got different accessions. We got different than the WA, but we got sterility. But for restoring that, we didn't have the restorers. The restorer which restore for WA didn't work for this. So whatever we identified was no use. So the problem for that is we don't have the restorers. So that's why we are still working. Maybe some other source of other than that what we identified, maybe something the natural population we may have restorer. Still that work is going. It should be done in both CMS and restorers. Restorer is a little easy. So once you convert, we can use. In case of CMS, first you have to improve the B line, then B line you have to convert into CMS line. So it takes a double. Like if it is finished in 4 years, a restorer breeding, it will take 8 years. We taken one irrigated restorer. We crossed with the donor for drought, which is having a drought and adaptability to DSR. Then actually it is having blast, drought, those genes are there. So then main thing is adaptability to DSR. That restorer we used with the CMS line. CMS is not bred for that. It is a normal irrigated CMS. But what the hybrid we got, because of heterozygous nature of the genes, whatever it is, it got the adaptability to DS. So for that, whatever hybrid released for irrigated situation, so we screened under the tolerance for the different stress in comparison with the varieties. Popular varieties also we took. So if you see both, hybrids are doing very good. We don't know what is the mechanism exactly involved. So that is not bred for this ecology or drought or whatever DSR. But what we see is it may be a heterosis or the buffering nature because the two parents are involved. So the buffering is more. So that is what that is yielding. Yield is compensated. If it is not bred for that, it will not give better yield. So that is even if you see in DSR or aerobic situation, even irrigated we are getting 6 ton. Exactly 6 ton should get in DSR also, but it will not happen exactly. So there is a little drought or little there irrigation problem, there will be yield penalty. So actual our objective is it should be as like of irrigated. Same 6 ton we should get. So that variety which is bred for the DRLH4 bred for DSR. So when you go in irrigated ecology also, it should be yield advantage. Here we are getting 5.5 ton in DSR. So when we tested in irrigated condition, we got 1 ton more. So irrigated, it will be always advantage. So there will be. So you will not get more yield than irrigated. Always. That is practically we have to accept. So, but it should not be the significance should not be more. There should not penalty should not be more. Those kind of lines we will discard it. There should be like a bench.